Greetings everyone, my name is Saibal and as you are already aware of, I am the current biology faculty from Nucleon Coaching Institute, and this is the continuation of our series that we have started, that I come and discuss about 4 topics regarding the entire biology that will be very helpful for you in your upcoming classes or upcoming exams, mostly for those students that are going to give the class 12 board exam on this 27th of March 2026. So please pay attention and let's make studying a bit fun. Okay? And these are some of my qualifications that you are already aware of by this point. And moving on, these are some of the chapters that we, or some of the topics that we have discussed before. Moving on further, we will be starting with today's new topic. Okay? Today's new topic discusses or is the one that I feel most connected to because of my background from MSc Plant Breeding and Genetics, mostly from genetics. And this is the part that deals with your genetics, which is this is the part that deals with our genetics, and the term that we use for it is pedigree analysis. From this flowchart itself, you will be able to understand that for males you represent it through a square, for females you represent it through a circle, and when there is a mating or a marriage between them, we simply make a line or simply connect the dots, or as to say, and make a connection. Consanguineous simply means that it is a very close form of a relationship. A close form of a relationship would be an incest form or an inbreeding form. That is mating between brothers and sisters, closely related brothers and sisters. Okay. Moving on to that, this is simply the crosses that you can observe. Okay, a cross between a male and a female would normally give you 2 males and 2 females. Okay. And this is what is represented. Now, you might think that what is the need of representing something like this? The basic need for representing a entire dichotomy of your generations through your pedigree is to give you the brief idea on what is the inheritance pattern for a particular disease, and for which I have given you a very good example of this autosomal recessive inheritance. Okay. Some of the autosomal recessive disorders that you are aware of and is present in your syllabus, the first and the foremost will be thalassemia. Okay, thalassemia is an autosomal recessive disorder wherein in the chromosome 11, okay, chromosome 11 and 13, you will find the deficiency or deletion of the genes that control your hemoglobin. Okay, hemoglobin A and hemoglobin B. Okay, the mutated gene will be represented through this and the unaffected parent. Okay, from a pedigree you can simply see that if there is a cross between an unaffected parent and a carrier parent is something or a term that we should be I will discuss it. Okay, a carrier parent is simply a parent that has the potential to pass the infected allele or the not infected allele, that would be something wrong to say. It is simply that the allele itself is defective, okay, and it has the capacity to be inherited in the subsequent generations without it actually showing itself in the current generation. That means if we talk about, and we have talked about, and you would have studied also, that something of a carrier parent, something of a carrier female, what that simply means is that the phenotype from the carrier progenies or the carrier individuals is not reflected in that current generation itself. It is simply that when a carrier parent crosses with a normal parent, they have the ability to transfer the phenotype and the phenotype to be expressed in the subsequent generations, okay, subsequent generation, and most probably if we talk about sex-linked inheritance, then it is mostly visualized in case of males as compared to females because of something called as dominance and a recessive relation between these alleles, okay. In this case, however, we are not talking about sex-linked inheritance, as to say, we are talking about autosomal linked or autosome linked inheritance. Now, what is this basically? Autosome linked inheritance simply says that there are, for example, if we talk about humans, there is 46 chromosomes. That means there are 23 pairs, out of which one pair is responsible for the entirety of your sex chromosomes or the sex organs. That means your entire sex organs are developed and controlled by those one pair of genes or one pair of chromosomes, but the rest of the 22 pairs form your entire body, that is your entire autosome, hence the name autosomes and your allosomes, okay. In this case also, since we are talking about autosomal recessive inheritance, remember it is talking about those 22 pairs, not the 23rd pair, okay. In case of humans, unaffected parent when crosses with a carrier parent, what happens is that in this case the chromosomes are normal, okay, as represented in these blacks. But in the case of a carrier, what happens is one of those chromosome pairs, okay, or one of that chromatid is mutated. That simply means that it is affected. Mutation in this case does not mean that there is some form of proper mutation, as to say, okay, mutation in the basis of either a base point mutation or a frame shift mutation. It is not simply talking about that, it is simply talking about the recessive nature of these alleles, okay. And what happens is that once they mate, there is a high chance or a possibility that this will be inherited, okay, or this will be inherited in the children of the subsequent generations, where one of those mutated genes will be actually transferred to the other, and as such they will always be affected. But what happens if those two carrier children are supposed to mate or happen to mate? What is going to happen? You will get an affected child, and this affected child can only possess this particular phenotype of the disease if and only if both of those parents were carried. And this is a case of a recessive one. Why? Because in a recessive form, remember, and you have studied it also, a recessive is a condition of the alleles wherein it is only able to express itself if and only if it is present in the homozygous condition. That means both of those have to be same. Hence, it will be only affected when both the chromatids are of mutated nature. Okay. This is what I meant to say from this small topic of pedigree analysis. And why is this important? Because this is going to help you to plan out, chart out whether or not your progenies can possess a particular genetic disorder, okay, because all of these are genetic disorders, and thalassemia is quite a deadly disease, okay. There are two forms of thalassemia, which is the thalassemia A and thalassemia B. Since we are not talking about thalassemia in detail at this point right now and sticking to pedigree, so this is what I will be leaving you with, okay. Moving on to the next topic, which is our vectors, cloning vectors. Although we have discussed how to integrate our vectors, but we have not discussed in basic sense what is this vector that we are talking about. What is this plasmid that we are talking about? Okay. Basically, a plasmid is an extra chromosomal segment of DNA with self-replicating and autonomous properties. Now, what does all these fancy terms mean? It simply means that these parts that are present in bacteria are able to replicate without the interference of the main bacterial genome, okay, or without main bacterial genome having to basically control or regulate their replication. This is an autonomous body. That means it simply replicates when and if it requires. Okay. And in that sense, what are we going to do is we are simply taking this example of a host and a chimeric. Okay, now what is this chimeric that you might say? Chimeric is nothing where we have actually added our part of the DNA that we wanted to integrate. Since we have already discussed on this, I am not going to explain it in too much detail, but I am going to explain how is this possible and what are these that you are seeing in your screen right now. Okay. This is called as an EcoR1. What is this EcoR1? This is a restriction site for the restriction enzyme EcoR1. Now what is this EcoR1? EcoR1 has been taken from E. coli, and R is simply the strain and 1 is the number. Okay, it simply is the nomenclature scheme, and every host or every plasmid contains some of these restriction enzyme sites. Now what are these restriction enzyme sites, you might say? These are nothing but palindromic sequences which are very specific to the restriction enzymes they carry over. Okay, and in that sense only, it will be specific onto where it starts and where it ends. That means a very specific portion will be cut if you add EcoR1, if you add Hins3, if you add BAM-HI, and if you add SA2. That means all of these are restriction enzymes. Okay, and this is the ampicillin resistance and tetracycline resistant genes. Okay, these will act as our markers. Why? Because I have told you that if you add a particular gene, that in its immediate outcome shows you an insertional inactivation if you had a marker in that particular place of cut out. Okay, if you cut out a part from your hand, okay, suppose if I cut out a part of my finger, okay, now I don't possess this index finger. Okay, and in that sense, the first thing that anybody will notice, okay, in a grafted, if I were to add another index finger, will be that there is a stitch mark. Okay, there is a stitch mark and it does not work like it should. This is what our basic purpose of marking a marker and the restriction sites for identifying whether or not our gene has properly integrated itself into the vector. Okay, like see, if there is your tetracycline resistance, okay, and if you cut out a part from this and add it on to the next part, there will be no tetracycline resistance in this part. Okay, and there will be a tetracycline resistance in this part, but there will be no ampicillin resistance. Why? Because we have simply taken out and replaced the ampicillin resistance with tetracycline resistance. So now, if you were to add ampicillin, this host or the host that is going to gain this vector will die if you put ampicillin. Okay, this is the basic idea on how to identify whether or not the vectors that we are taking have properly integrated the DNA that we are going to integrate into. Okay, I hope this is clear to all. Moving on to the next is our ozone layer depletion. Okay, now it is a very common term and yet a very scoreable part. Okay, so I would suggest that you don't miss it out or don't leave it in that arrogance that these are very easy chapters. Remember, if you are going to opt for, or you are going to opt for the most maximum marks that you can have rather than showing your intellect. If you want to show your intellect, then NEET is the best chance, or NEET is the best place to show it. Okay. If we move on to the ozone layer depletion, How does ozone layer deplete, you might ask? Ozone layer simply depletes through the action of CFC, is our chlorofluorocarbon, okay, or chlorofluorocarbon, okay. How does it form, or why is it recommended that you don't use any of those LED or any of those CFL bulbs, okay, or any of those old bulbs? Why? Because they release something called as CFC, chlorofluorocarbon, and what it does is that it binds with the ozone in the atmosphere, changing it to your O3, reacting with your nascent chlorine, which forms oxygen and removes the O3 part, and thereby depleting the layer of the ozone in the atmosphere. Now, why is this necessary, if you might ask? Why is this necessary? Why is this ozone layer important for us? Because this is the part that is going to reflect both your intra radiations and infrared radiations or ultraviolet radiations outside. It is the part that is going to keep you warm, or keeps our planet warm when there is no sun, to reflect and reflecting back the infrared radiation. And it is also the part that is responsible for reflecting the ultraviolet rays or the heavy cosmic rays from the other sources besides sun, okay. Sun also does that. Sun also radiates ultraviolet radiation, and that is also reflected back along with the cosmic radiations that is reflected to various other cosmic and heavenly bodies outside, okay. So see, in this there is UVA, UVB, that ozone forms the shield which protects you from UV rays, protecting you from melanoma. If you know what melanoma is, please write it in the comments, and I will be checking each and every comment of yours, because melanoma is a very common condition, and hence the reason why it is recommended that we take sunscreen, okay. What happens is that, see, in here there is your chlorine and fluorine, right? And where does this fluorine comes from? This fluorine comes from your CFC, that is your chlorofluorocarbon. What happens in the first case is ultraviolet rays split the chlorine atom away from the CFC, that is chlorofluorocarbon, which what happens is that the chlorine adds to, or chlorine binds to the ozone, that is O3 molecule, changing it into your O2 and releasing a nascent oxygen, okay, leaving behind a nascent oxygen and causing this ozone depletion. Now, where can you find your CFCs? You can find it in your aerosols, refrigerants, solvents. HCFC, hydro, chloro, fluorocarbons, you will find in aerosols, refrigerants and solvents as well. Hence, it is used, or hence it is recommended or advised, not recommended, advised that you don't use too much of AC in these summer days. Why? Because these ACs, these refrigerators, these appliances, they rely on CFCs and release a heavy amount of CFCs, which in fact is the reason that your ozone is relatively depleted as compared in the 100 years or the 200 years that it has been detected, okay. So think about the environment, okay, and try to minimalize it, okay. Try to minimalize your usage of these appliances and make it so that you can keep it for your future generations also. Since you are the future, I would expect, or it is my humble request, to take care of the planet that you will inhabit and you will inherit. This is your planet, this is your world. We are also there, but our time is very limited as compared to yours. You are the ones that will be inheriting this country, this world, entirety of this, and it falls under your jurisdiction to protect it by the means necessary. Okay. Enough of logic. Okay. Moving on. What do you see in this? What do you understand in this? Before I say what is this is the TS section, transverse section of the spermatogenesis process along with the spermiogenesis process in the seminal vesicle itself. Okay, this is also a very important topic. Okay, this is also a very important topic, as in you will see in these pictures itself that it has been mentioned. What is this? That this is the mitotic division of the spermatogonium. Second will be meiotic division of the primary spermatocyte, and third will be the meiotic division of the secondary spermatocyte. Okay, starting from this phase. Okay, in that first, these are called as spermatogonium, which divide into the first meiotic division, as you can see, first meiotic division, causing it to be haploids. Okay. Haploids and a primary spermatocyte that is formed, which releases the other bodies to be carried over and to develop into secondary spermatocyte through another round of meiotic division, forming our spermatids. Okay, these are what? These are forming of the spermatids. What happens in this? That you can see that these are spermatogonia, these are the Sertoli cells which are responsible for maintaining or basically nourishing the developing spermatids in these sections. Okay, from your primary spermatocyte moving on to secondary spermatocyte, and this process continues. Okay, this is a layer. Okay, what happens is that from the layer of these sections you will find the spermatogonia, which meiotically divides into form primary spermatocyte, which then again meiotically divides to form your secondary spermatocyte, which then divides mitotically to give you your spermatids in a higher count, and these are developed in these lateral section of your seminal vesicles, moved over to your epididymis and stored for about 3 to 72 hours. 3 days is 72 hours. Okay, if by then it is not released or if it is not used, they are simply broken down and the process again continues. Now, what is this spermatogenesis and what is this spermiogenesis? That is a question that I will ask I would expect you to give me an answer from this diagram itself in the comment section, and I will be checking it, and I hope that this video was helpful to you in understanding these 4 chapters along with their weightage and along with the possible questions that can be asked. Okay, and I hope that you continue to watch this series, and I hope to bring you out with more better videos and more better topics to cover, along with my own assessment of the exam papers that I have been preparing for your boards as well as for NEET. Okay, I wish you all the best and thank you for listening to me.